Welcome back to Anatomy and Physiology on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. This playlist, as I mentioned before, is going to be dedicated to talking about gustation and olfaction, which in layman's terms is taste and smell. We've already talked about gustation. In this part of the playlist, we're going to be discussing olfaction or smell, and we'll be talking a lot about the physiology and the signaling, and hopefully we'll learn enough to be able to answer this question, which is how, based on smell, would I distinguish between something that smells really good, like this pizza, and a skunk, which I think we all know they smell terrible, at least their musk does. And the cool thing about the answer to this question right here, how do we distinguish between these things, it actually won a Nobel Prize. Okay, so let's actually get into the anatomy first. All right, so obviously when we smell, we're using our nose, right? So here's our nasal cavity right here. Of course, our brain is up here in the cranial cavity, and if you think about the structure of the cranial cavity, it's surrounded by bone. And if we look at this bone that actually separates the nasal cavity from the cranial cavity, that's actually this bone right here, this bone actually contains something called the cribriform plate. And if you go back and look at what you probably learned back in bone physiology, or at least the anatomy, you hopefully remember at least mentioning the cribriform plate. Now, right above the cribriform plate, we have this structure called the olfactory bulb. In fact, there's a left and a right olfactory bulb. When you covered the brain in lab, you probably had to identify this structure. Now, beneath the cribriform plate, especially down here in the gray region, this is actually the nasal cavity. And what we see flanking the nasal cavity is we have a bunch of epithelial cells right here. And interspersed in those epithelial cells are chemoreceptor cells. These chemoreceptor cells are basically the cells that detect smell. Okay, so these are our sensory neurons right here. We'll talk about those in more detail in a little bit. But it suffices to say that these sensory neurons, which are chemoreceptors, in the nasal cavity side, they have these little hair projections called cilia. It'll turn out that the molecules that we smell actually bind to parts of these cilia. And if we follow these chemoreceptor cells upward in between these epithelial cells, what we'll see is they'll actually move through these gaps in the cribriform plate. And their axons will actually synapse with these cells up here called mitral cells. Now the mitral cells are the first cells that we encounter in each of the olfactory bulbs. If we look at these mitral cells right here, of course they're not labeled, we see that their axons actually project toward the brain. And all these mitral cells, their axons will converge into one nerve, which is the olfactory nerve, or cranial nerve number 1. Okay? So hopefully you understand this setup a little bit. Now, these little blue dots right here are molecules that we can smell, and scientifically speaking, we call them odorants. So in order to detect a molecule, to smell it, the odorant has to do two things. One, that odorant has to be dissolved in mucus in the nasal cavity. So I guess it goes without saying, of course, the odorant's going to have to enter the nasal cavity, and there it will dissolve in mucus that we have in the nasal cavity. The second thing is the odorant is going to have to bind to an odorant receptor. So here's an in-depth, or I should say a zoomed-in look at one of the cilia on these chemoreceptors. So we have different receptors here for the odorant, so they're called odorant receptors. And the odorant would of course have to bind to the odorant receptor. And only then will a person be able to detect that molecule and smell it. So odorant has to be dissolved in mucus, and then it has to bind to these receptors on the cilia termed odorant receptors. Okay, so let's actually look at the signaling for these odorant receptors right here. So here's a receptor. It's a G protein coupled receptor, meaning it's a transmembrane receptor that is coupled to this G protein right here. So here's a specific odorant. The odorant will bind to the odorant receptor. That'll cause changes in conformation which activate the G protein. So here's our G protein, which is called G sub olf. This is actually olfactory, so it's a specific G protein for smell. And of course, through a series of processes, the G protein, that is the alpha subunit, which is G olf, picks up GTP, and when it picks up GTP, it will move over and bind to certain enzymes. The enzyme that the G protein binds to in the case of olfaction is adenylate cyclase. And when the G protein binds to adenylate cyclase becomes activated. And it will start converting adenosine triphosphate, or ATP, into cyclic AMP. Now this cyclic AMP is our second messenger here. So cyclic AMP will go around the cell, and one of the proteins it binds to and activates is a cyclic AMP gated ion channel. And so whenever a cyclic AMP binds to this protein and it becomes activated, you'll allow ions to start influxing like sodium and calcium. And since both of these ions are positively charged, they're cations, when they influx it results in depolarization of this cell. Okay? And so all of this process occurs only when an odorant binds to that odorant receptor. So in other words, whenever these odorants bind to particular receptors on the cilia of these chemoreceptors, that will cause overall the chemoreceptor to become activated, and it will start firing nerve impulses toward the mitral cells, which are located in the olfactory bulb. Okay, so hopefully that makes sense. Hopefully the signaling makes sense. Now, the question is, and this relates back to this original question that I posed, how do we differentiate between something that smells good and something that smells bad? Or rather, how do we just distinguish between different smells? Okay. Now, I'm going to show you this picture right here, and it refers to something called olfactory coding. And it looks very complicated, but let me kind of break this down. All right, so if we think of all the possible molecules in the environment that we could possibly smell, there's, for the sake of argument, practically an infinite number of potential molecules, or at least a ridiculously high number that's unfathomable. So now let's move on to this slide. This slide is describing something called olfactory coding, and actually the discovery of this is what led to a Nobel Prize. And this figure up here in particular looks pretty complicated, but actually it's a fairly simple concept and kind of cool. So we've got an infinite or potentially infinite number of odorants. I mean, these are all different looking molecules and they all smell different. So how do we potentially differentiate an infinite number of odorants and their smell? Do we have an infinite number of odorant receptors? Remember, here we have an odorant binding to its receptor. But are there an infinite number of receptors for an infinite number of odorants? And the answer is no. And that's going to lead us to our first key point here. All odorant receptors have broad specificity. So what that means is we do not have a one odorant, one receptor situation. What we have instead is we have a finite number of receptors, and each receptor can bind many odorants. Okay, so these receptors have broad specificity. So for example, I mean, this is just an arbitrary number I'm throwing out, but maybe this receptor can bind thousands of different odorants, and it can bind them to different extents. But we don't have one odorant and one receptor for every odorant. Okay, each of these receptors has broad specificity. Okay, and key point number 2 is really what won the Nobel Prize. The combinations of activated odorant receptors dictates which smell is detected. Okay? So in this experiment, and I believe this is actually experimental data that they used, they had a bunch of different odorants here which actually smell different. And they had 14 odorant receptors. So we're going to just number them receptor 1, 2, and so on and so forth. So let's actually look at compound A right here. This is odorant A. This odorant bound to an activated odorant receptor number 5, okay? And this odorant smells rancid, sour, and goat-like, whatever that means. But now let's take a look at odorant C, okay? Odorant C also activates receptor 5. However, it also activates 1, 4, 7, 10, and 11. We could also look at odorant B. This one activates odorant receptors 2 and 6. And so what we see here is key point 2. The combinations of activated odorant receptors dictates which smell is detected. If you had a situation where each odorant had a specific odorant receptor, meaning every single odorant only bound to one odorant receptor, and you had one odorant receptor for every odorant, you would have to have a genome that's probably 1,000 times the size of what we have, maybe even 1,000,000 times. Okay, that would be absolutely absurd. So to simplify things, what happens is each odorant binds to either one or more odorant receptors, and that combination of receptors actually allows us to differentiate the different smells. So look at odorant receptor 5. There appears to be a lot of different compounds that bind to receptor 5. However, each one of those has a different combination of other receptors that they bind to. Okay? And if you look at all these compounds, they certainly look different. All right? And that's how we differentiate different smells. And this combination of different odorant receptors being activated is called olfactory coding. And then key point number 3 here, which is not as important as this, but it's also kind of interesting, is that the olfactory bulb is organized by the type of compound detected. Okay? So let's take a look at this. So here we have the rostral side on the left, caudal side on the right. Then we have dorsal, lateral, ventral, medial, dorsal. Don't really worry about what these regions are, but if we look at the colors over here, like this blue, light blue region right here, this region of the olfactory bulb seems to detect aliphatic hydrocarbon chains. If we look at this part here that's kind of a reddish, kind of a dark red, this area is going to detect highly water-soluble compounds. This part that's purple, or purple parts right here, this would detect methyl-substituted bicyclic compounds. Okay, this is some heavy-duty organic chemistry stuff right here, but hopefully what you see is that different regions of the olfactory bulb seem to detect or have a propensity to detect different types of organic compounds. And most compounds that we're detecting are organic compounds. Okay, so key points here. All odorant receptors have broad specificity. They can bind multiple odorants. Also, the combinations of activated odorant receptors dictates which smells detected. Okay? That allows us to be able to detect a much larger range of compounds, but also allows us to have a smaller number of odorant receptors. Okay? And then also the olfactory bulb is organized by the type of compound detected. Okay? This whole thing is called olfactory coding. Now, the very last thing I want to talk about are the olfactory pathways to the brain. We've already taken a look at this right here. Here's our olfactory bulb up here. This is our cribriform plate, and we have our mitral cells. Notice we have different colored mitral cells here. That's basically just representing the type of compound that is detected. But in any case, the axons of all these mitral cells are going to converge and become the olfactory nerve, which is cranial nerve number 1. And this nerve is going to project posteriorly toward the brain. And when it gets to the brain, it has 3 major places that it's going to go. The first place is the olfactory cortex. Okay? This is basically where we're going to have conscious perception of smell. If you were to damage the olfactory cortex, you would no longer be able to smell. And going along with this would be the olfaction association area, which would allow us to associate a specific smell with a name, let's say. So for example, you know that something is a pepperoni pizza versus a skunk musk. All right. The second place that the olfactory nerve sends this information is the hypothalamus and amygdala, both of which are a part of the limbic system. And this is going to yield motivational and emotional aspects of the smell. So the example that's often used of this is, for example, if you have a late family member, let's say like a grandmother, that at one point baked a certain type of cookie that has certain smell. If you were any other place and you smelled that smell, it might conjure up emotions that relate to your late grandmother or something like that. Not to be depressing or whatnot. Okay, and the third place is the hippocampus. The hippocampus is also part of the limbic system, but it just deals with strict memory. And so if you have memory of the smell, when you encounter that smell a subsequent time, you'll be able to remember that smell, and along with the olfactory association cortex, you'll be able to associate that smell with an idea or a name. Okay? So hopefully this video gave you a good understanding of the olfactory system. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.